Good morning, it's August 25th, and this is your daily brief in science. Here's everything you need to know. Clearmind Medicine has announced the U.S. publication of a patent application for a combination of MDMA and N-acylethanolamines, notably palmitoylethanolamide, or PEA, to treat PTSD, anxiety, and eating disorders, developed with Neurothera Labs, Inc. The filing arrives as the FDA issued final guidance in mid-July 2026 on psychedelic drug development, including MDMA, and outlining regulatory considerations for clinical investigations. Embodiments suggest PEA could boost MDMA's potency, enable lower dosing, reduce side effects, and widen the therapeutic window, potentially strengthening both clinical and commercial prospects. The market reacted modestly, with CMND trading around $1.72 on elevated volume amid the IP news and regulatory developments. The Form 6K includes the press release text and notes that the patent is not yet issued, outlining current status, while the release provides FAQs about the patent, potential benefits, portfolio details, and cites Globe Newswire for the full press release. Clearmind's shares trade on NASDAQ under the ticker CMND, and the press release contains standard forward-looking statements with risk disclosures similar to those in SciSpark filings. Key risks highlighted include patent validity, the possibility that potency or tolerability is not enhanced, regulatory challenges, MDMA's Schedule I status, funding needs, and reliance on third parties. Forward-looking statements acknowledge that patent issuance is not guaranteed and benefits depend on future developments amid regulatory and clinical uncertainties. Exhibit 99.1 provides background on the MDMA N-acylethanolamines IP and its potential regulatory and market implications, with the release underscoring potential regulatory and development advantages from the FDA guidance and an expanded IP portfolio. A global heat spike in the oceans set a new record, with surface temperatures around 21.1 degrees Celsius, and experts say this peak could rise further in the coming months. The rise is attributed mainly to long-term human-caused warming from greenhouse gas emissions, amplified by the current El Niño in the tropical Pacific. Beyond the heat, marine ecosystems face stress that damages coral reefs and seagrass meadows, disrupting food chains and affecting fisheries and aquaculture. The report also highlights the role of science journalism in informing the public about discoveries and climate-related issues, including Scientific American's coverage. Leaders are calling for rapid policy action to cut fossil fuel emissions and implement conservation measures intended to protect at least 30% of the planet by 2030, with on-the-ground commitment and support for indigenous peoples and local communities. The piece notes Pravda.ru as the source, with Anton Kulikov as author and news correspondent for the English edition. Scientists warn these changes could have ecological and economic consequences, including risks to global food security and livelihoods tied to fisheries. Officials and advocates are pushing for accelerated investment in clean ocean energy such as offshore wind and stronger protection of marine and coastal ecosystems to mitigate impacts on fisheries. Copernicus emphasizes that the record highlights broader climate impacts and urges attention to regional consequences, with explanatory context and potential effects shown in a companion video. Reporting draws on outlets including The Guardian, The Washington Post, NOAA, and The Independent to corroborate the record and discuss ecological and food security implications. Overall, the record underscores ongoing climate change impacts on the marine environment and the risks to coastal communities and ecosystems, with warming extending from the Atlantic near Ireland to the Mediterranean across Europe, driven by climate change and fossil fuel burning. A yeast-based manufacturing platform aims to replace the traditional egg method for influenza vaccines by using M2-containing virus-like particles that are non-infectious and designed to elicit broader protection across subtypes. The approach focuses immune attention on the conserved M2 protein, potentially enabling cross-subtype immunity rather than strain-specific protection. The VLPs display abundant M2 on their surface, mimicking the virus without genetic material to boost immune recognition. Funding comes from the University of Michigan College of Engineering and the NSF Career Program, with results presented at ACS Fall 2026 in Chicago. Future work will assess durability and breadth of immunity, identify the best immune mechanisms, and evaluate efficacy in humans while exploring dose control, stability, and safety for potential oral formulations. The platform envisions moving from injections to oral vaccines via fermentation technology, with ongoing findings shared at the ACS Fall 2026 meeting. The study, supported in part by an NSF Career Award, was presented August 24 at ACS Fall 2026, highlighting potential single-shot long-term protection against multiple flu strains. Asparavax has licensed the yeast-based technology to develop an oral vaccine, signaling interest in broader applications and progression toward human testing. Results are preliminary and preclinical in mice, with questions remaining about durability, human effectiveness, and suitability for immunocompromised individuals. In mouse studies, M2-VLP vaccination produced antibodies against multiple strains and provided 100% protection against 3 tested strains, indicating potential broad efficacy. Next steps include determining how long immunity lasts in animal models and advancing toward clinical testing while refining production methods for rapid pandemic response. A new study from Texas A&M, NASA's Johnson Space Center, and Purdue University lays out a practical framework to manage traffic around Gateway, the lunar outpost planned to orbit in a near-rectilinear halo orbit. Researchers built a computer-simulated toolkit to create a mathematical flight manual for safe multi-vehicle operations in cislunar space. They introduce a string-of-pearls approach, lining up arriving vehicles in timing and spacing relative to Gateway to enable orderly docking and departures. Loitering, defined as controlled holding patterns near specific orbits, helps sequence landings and departures while conserving fuel. A relative orbit maintenance algorithm tracks each incoming or outgoing vehicle against Gateway, enabling tighter formations and cheaper course corrections. The work, published in Acta Astronautica in 2026, shows that small increases in station-keeping maneuvers can greatly improve predictability and safety, with only a modest rise in propellant use. This approach aims to balance safety, fuel efficiency, and operational demand in the NRHO environment around Gateway. Managing multiple simultaneous lunar missions will require new orbital maneuvers and queuing strategies to keep spacecraft loitering near Gateway while accounting for thruster issues and navigational errors. The central idea is to lay mathematical rules of the road for lunar traffic, delivering a safer and more efficient workflow for both crewed and robotic missions. Diane Davis of Texas A&M says traffic management is as crucial as propulsion design for sustainable lunar exploration, and aims to train the next generation of space traffic engineers. The study portrays a future where robust traffic management becomes a core pillar of space infrastructure, shaping how lunar exploration proceeds for decades. A look at immunometabolism shows how T and B cells reshape their energy use during autoimmunity. In MS and related models, pathogenic Th17 cells shift toward glycolysis, increasing GLUT1, HK, GAPDH, PKM, and LDHA, and blocking glycolysis can reduce CNS inflammation and demyelination. Dysregulated glycolysis and mTORC1/H-IF1 alpha signaling push CD4 plus T cells toward Th1 and the 17 responses while suppressing TREG differentiation, with glycolysis inhibitors like 3-BPA, 2DG, and PFKFB3 inhibitors shown to lessen disease in models and boost TREGs. The adaptive immune system relies on T and B lymphocytes, and autoimmunity arises when self-reactive cells escape tolerance mechanisms. Memory T cells favor oxidative phosphorylation, with higher mitochondrial content and elongated mitochondria, supporting rapid responses upon re-exposure to antigen. Immunometabolism links lymphocyte function to metabolic programs, and disrupting these pathways can fuel autoimmune activation and disease progression. A comprehensive view shows metabolic reprogramming in T and B cells shaping the onset and progression of T-cell-driven autoimmunity, with glycolysis, the mTOR/HIF1 alpha axis, GLS1, and DHODH as promising therapeutic targets. Effector T cells rely on glycolysis and de novo fatty acid synthesis, whereas regulatory T cells depend more on fatty acid oxidation and OXPHOS, highlighting metabolic distinctions that influence helper versus regulatory outcomes. Autoimmune diseases such as MS, Type 1 diabetes, rheumatoid arthritis, and SLE involve dysregulated T-cell mediated responses, including cytotoxic T-cells, pro-inflammatory CD4+ T helpers, and interactions with APCs and B cells. Mitochondrial dysfunction in CD4+ T cells, altered ROS, deficits in SOD/GPX, and impaired apoptotic clearance contributes to MS progression by supporting autoreactive memory T cell survival and inflammatory signaling. Restoring apoptosis pathways can mitigate disease in models. MS therapies like glatiramer acetate, IFN-beta, and DHODH inhibitors modulate T-cell metabolism by reducing glycolysis and oxidative phosphorylation, aligning patient T-cell profiles closer to healthy controls. B-cell metabolism shifts from resting OXPHOS/FAO to glycolysis upon activation. Germinal center B cells favor FAO in the dark zone, with glycolytic bursts upon TFH help to re-enter proliferation, while plasma cells rely on high OXPHOS and de novo fatty acid synthesis for antibody production. Naive T cells predominantly use OXPHOS. Activation reprograms them to glycolysis, fatty acid synthesis, and glutaminolysis to support proliferation, with CMYC driving upregulation of glycolytic genes such as GLUT1. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.